So, biotechnology application, one of the easiest step. You don't face issue in this. That's why my purpose is to make it perfect. Biotechnology, we learned in the previous chapter, deal with the industrial scale production, large scale production of what? Large scale production of medicine, pharmaceutical, genetically modified organism, fungus, plant, or animal. Basically, genetically modified organism in simplest way. Correct? Application, there are a lot of application in the medicine purpose, in diagnosis, in crops for agriculture, in the processed food, bioremediation. Bioremediation means to control waste treatment and energy production. Correct? Three important critical area of the biotechnology where we did good job and we have to provide best catalyst in the form of either better organism or pure enzyme. Okay, so we have to provide best catalyst, effective catalyst. Second, any reaction we are doing, there should be perfect condition to the engineering process. So this catalyst what we produce can act properly. So first, best catalyst in the form of pure enzyme or microbe. Second, condition to act this catalyst, and finally to get the final product. To get the final product, so we can get final protein or organic compound. That the benefit of the biotechnology is food production increase and health is better now among the people. Right? What are the three options by which we can increase food production? Simple. Chemical use, so you can increase production. Organic agriculture, and last is genetically modified crop. Correct? Three options we have. Okay. Next, green revolution. In case of green revolution, we tripled the food supply. This is important word. How many times food production increased after green revolution? 3 times. But not enough to feed the growing human population. Not sufficient. Increased yield. What was the reason for the increased yield? Is due to the better crop quality. We have better crop variety now. Correct? हाँ.
हमें थोड़ी देर में कैमरा
क्या?
2 मिनट के बाद कैमरा कर
ठीक है. आदित्य तुम अच्छा. Next. But crop variety improvement is one of the reason, partly. Main reason is the better management practice we have, and this is dangerous. Use of agrochemical product like fertilizer and pesticide. This is the main reason. Better management and this. Better management is a good thing, but agrochemical is dangerous. Correct? Why? Because one thing, it is very expensive. Second thing, uh, there are a lot of harmful effect. Correct? Disease. Correct? So further increase in the yield with the existing variety are not possible by the conventional method. Whatever we increase variety, we cannot further increase if we use old method. We have to use new method. As traditional breeding technique fail to keep the pace of the demand. Old technique. Traditional means old technique cannot manage everything. And to provide the sufficiently fast and efficient system of crop improvement, we don't have. But we got new technology of tissue culture. Correct? In during 1950s, whole plant could be regenerated from the one small part. Example, you have one small plant, and this small part of the plant, example leaf, can produce the complete plant. So this leaf will be known as explant. Explant means any part of the plant is grown in the test tube under the perfect condition in the special medium, special nutrition. So what is happen? Example, I take a root of a plant. Okay, I put in the test tube. I make sure there is no contamination. I provide proper nutrition to this root. What will happen? Ultimately, it will form the complete plant. So this small part from which we can make full plant is known as explant. And capacity to generate the complete plant from the any plant is the potential. Totipotency. Totipotency means ability of a plant to produce a complete plant. Correct? In this case, when we provide nutrition to the plant, nutrition medium, this nutrition medium should have the carbon source. What should be the carbon source? Sucrose. Why? Energy. Correct? While we provide nutrition, there should be inorganic salt, electrolyte, vitamins, amino acid, and growth regulator like oxygen. Very important. Which growth regulator is really, really important? Oxygen and cytokinin. Why? Oxygen responsible for root growth. Cytokinin is for the stem growth or you know remaining all shoot system. Cytokinin for shoot system, oxygen for the root system. Correct. So hormone are compulsory, vitamin, salt, amino acid are compulsory, sucrose is compulsory. Question can be asked: what should be the component of nutrient medium should be present when you want to grow the plant? Correct. What is the benefit of tissue culture? We can produce large number of plant in a very short duration. Very short. Okay. And this method of tissue culture by which we can produce thousands of plants even in a day is known as a micro propagation. Propagation where we can produce large number of plants in a short time. Correct?
Then, sir, that is called tissue culture, no?
Yes, Ryan.
So then that is called tissue culture, no?
By tissue culture, we are propagating at the larger scale. Okay. And in this large scale, all plants are exactly same to original plant. If, for example, you take X plant, you use X plant in the tissue culture, you produce thousands of plants. All plant will be same as X plant. Okay. Soma colon. Soma colon means exact copy. Exact copy. They are exact copy of each other. Many plant like tomato, banana, and apple are produced by using the commercially used by using micropropagation. Question can be asked: ki, what are the method what are the plant can be produced by micropropagation? Tomato, banana, apple. Tomato, banana, apple. Correct? Tomato, banana, apple. Who is this Devans? Okay, next. ये तो common statement कि visit any lab to understand this better. You should visit. Next. Another important application of this method is recovery of the healthy plant from the diseased plant. Even if plant is infected with the virus, very, very, very important. Meristem, apical meristem or axillary meristem are never infected by the virus because these cells divide very rapidly. So virus cannot infect the meristem part. Correct? So even if plant is damaged, plant is uh, plant got a disease from some virus, still you can get healthy part of the plant, and this healthy part is meristem. Okay. So if we remove meristem and grow in the lab, we can obtain pure virus free plant without any virus. If you take meristem and grow in lab, you can make full plant. One benefit. Second benefit, plant will be free of any virus. Correct. And by using this, scientists are able to culture meristem of banana, sugarcane, and potato. So tomato, banana, and apple for somacolon, micropropagation. And virus-free plant we are able to make in case of potato, banana, common, and sugarcane. Okay, next. So many scientists isolated the single cell from the plant. Okay, and after digesting the cell wall with the cellulase enzyme, they got isolated naked protoplast. Naked protoplast means a cell that have plant cell that have plasma membrane but no cell wall. Plant cell without cell wall is known as protoplast. Okay, protoplast is the plant cell without cell wall. Plant cell without cell wall. Cell wall. Next. Isolated protoplast of two different variety of plant is having desirable character can be fused to get hybrid protoplast, which can be further grown in the new plant, and this is known as somatic hybrid. And best example, Ryan, right? Somatic hybrid. Suresh sir, somatic hybrid
Yes.
example, if you take tomato and potato, first have their protoplast, fuse them in the lab, fuse their desirable character, and you will get pomato, which is a mixture of tomato and potato, and have both the property. Though this concept was not really successful, but at least you are trying to do new experiment. Okay, tomato. While the process of the making hybrid is known as somatic hybridization. Correct. The hybrid is somatic hybrid, and process is somatic hybridization. Okay. Imagine a situation when protoplast of tomato fuse with the potato and then grown to form new hybrid plant that have property of both tomato and potato. Well, this has been achieved, and we formed what, Ryan? We formed
Tomato.
tomato. Unfortunately, this plant did not have all the desirable character what we expected, but at least we tried. Okay, we are able to make tomato. Next, any other path in the genetics where farmer can get the maximum yield? Answer is yes. We can reduce use of fertilizer and chemical because we know there are a lot of harmful effect of the fertilizer. How? We can use GMO, genetically modified organism. Which organism can we use? Any plant, bacteria, fungus, animal, anybody. Gene can be changed. That is genetically modified plant. Correct. What is the benefit of having genetically modification? First, plant can tolerate more stress, more cold, more drought, more salt, more heat. They can tolerate more. Reduce resilience on the chemical pesticide. We don't need to use pesticide now. Pest resistant plant we produce. Third benefit, first class में लगा था and class हम्म, तो हम लेकिन भाई बात नहीं बहुत गलत है। I want to tell you a very short story in 1 minute, guys. I just got a call from a coaching institute. Okay, like he's friend of mine. He asked me, "Ki, what are you doing?" I said, "Ki, I'm taking class." And he said, "Ki, तूने तो ऐसे ही पंगा ले रखा है यार, 12-12 घंटे कौन पढ़ाता है?" This statement is given by the coaching institute owner कि 12-12 घंटे कौन पढ़ाता है. 3-4 hour is okay, but 12 hour every single day, 10 hour, 8 hour doesn't make sense. And I was shocked कि उनको तो अच्छा लगना चाहिए ना कि हाँ student are getting result. Some like they will do better. You know, because commercially it's not good. Commercially, if everybody is doing this, then coaching institute will get more responsibility. They have to have children for 12 hours. So they cannot afford because they have to give double salary to the teacher if teacher work for 12 hours, right? So that is the thing. Come to the point. Very popular, all these things. Third, we will reduce post-harvest loss. Once you cut the crop, sometimes there is a loss because of they cannot, in the end, there is tufan, there is flood, there is unnecessary too much light or less light. So this post-harvest loss it will reduce. It increase efficiency of the plant to use more nutrient from the soil. Correct. This prevent early exhaustion of the soil fertility. Per plant can properly absorb all the nutrition what is required. So soil fertility will not be lost suddenly, like too early. And last, of course, nutrition value golden rice have vitamin A. This is very popular question. In addition to these, GM crop are used create tailor-made plant, and this already ready-made plant is good source for the industries in the form of like they can provide more starch, they can provide fuel, they can provide pharmaceutical. In those industry, in starch, fuel, and pharmaceutical industry, we need tailor-made plant already prepared. Only this crop have more carbohydrate. Correct. If you want to make more alcohol, this is the best. Okay. Application of the biotechnology in agriculture will is plant making plant resistant from the pest. Correct. So we have to use less pesticide. Example, Bt toxin. In case of Bt toxin, we use the bacteria Bacillus thuringiensis. This Bt toxin gene are cloned from the bacteria, correct, and expressed in the plant to provide resistance to the insect without need of the insecticides. Correct, resistance to the insect without need of the insecticide. So we don't need to spray any insecticide. Still, we can kill the insect. And this is a kind of biopesticide. We are killing the pest, but we are using living organism. We are not using chemical. So we made BT cotton, we made BT corn, we made rice, tomato, potato, and soya bean. Very, very important. What are the crop? What are the biopesticide crop? Cotton, corn, rice, tomato, potato, and soybean. BT cotton. Very popular question from next two topics. We use bacteria Bacillus thuringiensis. Bacteria produce protein. This protein kill insect. Example: if it kill leptopterans are armyworm or budworm. Coleopteran, beetle. Dipteran, flies, mosquito. So Bacillus thuringiensis can kill all these things. Correct? Like Bacillus thuringiensis can kill butterfly larva. Can kill butterfly larva. Dipterans, coleopterans. Coleo means beetle. Leptopterans, budworm or armyworm. These Bacillus thuringiensis form protein in special phase of their growth. Correct? And these crystals have toxin, insecticidal protein. So this protein is insecticide. This protein will kill the insect. But why this protein is not killing bacteria? Because it is present in the inactive form. Because it is present in the inactive form. Once it enter into the insect, it will become active. Why? Because gut have alkaline pH, and because of the gut pH, this toxin will become active and it will solubilize the crystal. Active form of the toxin will solubilize the crystal. Toxin will bind to the midgut epithelial cell, very important term. Midgut epithelial cell, create pore, cell swelling, breakdown of the cell, and eventually cell death. Okay, so what we are doing, we know bacteria can kill insects, specific insect. What bacteria have one dangerous chemical for insect. So, but this dangerous chemical is not killing bacteria. Why? Because this dangerous chemical activate only in the insect gut due to the alkaline pH. Due to the alkaline pH, gut, especially in the midgut of the epithelial cell, there will be pores, swelling, lysis, and death of the insect. Right? So we have to obtain a specific Bt toxin gene from the bacteria and enter into the plant like cotton. Which gene we will choose? Gene will depend upon the nature of crop and which pest are we killing. Correct? So this is very insect specific. Example: toxin encoded by gene cry1AC have this nature cry gene cry1AC cry gene. There are many. Example: 1AC 2AB are used to kill cotton bollworm and 1AB control corn borer. Okay. So very important. By the way, in Haryanvi there is a word abbi. Abbi means, who is Haryanvi here? Even I am not able to explain this properly. Anybody who is from Haryana? No one. Okay. So abbi means a person who create nuisance all the time. Very,
एबी ABBY।
हाँ abbi,
हाँ समझ गया, मैं समझ
है ना?
हाँ
abbi means जो सारा दिन ऐसे लड़ाई झगड़े या कुछ ऐसा करने का शौक
हाँ,
रखते हैं
वो
कि कुछ
बोलते हैं ना,
तूफानी
एब
करना
एब भी है बहुत ज्यादा, ऐब नहीं होने चाहिए।
है। ऐब नहीं होनी चाहिए किसी इंसान में ना। ऐब नहीं होने चाहिए, इसका मतलब वो बहुत ज्यादा वो नहीं होना चाहिए, कुछ नेगेटिव कैरेक्टर नहीं होने चाहिए। दैट इज द मीनिंग ऑफ एबी। सो फ्यू ईयर अगो आई मेड दिस ट्रिक। एक एसी को ठीक करने के लिए दो एबी बुलाए और उनमें से एक एबी चोर है। Okay. To repair one AC, we called two Abby, and one of the person is thief. Okay. What does it mean? One AC and two AB control bulai. Bulai means bollworm. Bulai bollworm. And one AB is used to control corn borer. Corn. Understood? Because this is popular question and you make mistake in this. Okay. As you can see here, a cotton ball destroyed by bollworm in the green color, and second fully mature cotton ball. Correct. So BT cotton is done. Tell me, guys, in this topic, you face issue in this cry 1 AC, cry 1 AB, cry 2 AB. What is the role of each? How many of you face issue in this? Raise your hand. No one. Leon. Okay, Nirzila, Rayan, Aditya. Because I used to face issue. When I see in exam, and there are like, I feel which one? Okay? Or you can remember ki, one AB is corn borer, remaining are bollworm. One
So that's
AB
how I
is
have
corn borer,
learned it.
right? So this is one of the easiest way. Yeah, this is right way, Anvita. He just focus on one remaining, one AB is corn borer, remaining is bollworm. Next, pest resistant plant. We have lot of nematode parasite that can affect plant and animal both. Correct? They can affect human being also. Same nematode, Melodygyne incognitia. This nematode affect the root of the tobacco plant and reduce the yield production. A novel strategy was adopted to prevent this infection infestation, and this process name was RNA interference. We used RNA interference process. Correct. This RNA interference process occur in all eukaryotic as the cellular defense. The same mechanism is naturally present in the eukaryote. In this case, we silence the specific mRNA because we make complementary, like RNA is present. That's why we'll make double-stranded DNA. We will make double-stranded RNA, and if you have double-stranded RNA, translation is not possible. So this mRNA will be silenced. Example: we have one melodic incognitia DNA. This DNA will produce RNA. Okay, this is double-stranded DNA. It will produce single-stranded RNA, and this RNA in future will make harmful protein which can cause disease. So what body will do? We will make complementary of this. We will make double-stranded RNA. Due to double-stranded RNA, protein formation is not possible. If protein formation not possible, no harmful thing. Right? No harmful thing. How to get this complementary RNA? We can obtain from the virus that have RNA, or we can use mobile genetic element like transposons. Okay? These are the two sources, because in both the case RNA virus or transposons, they replicate using RNA intermediate. So we can use their RNA. Correct? We can obtain RNA from them because they use. We are using agrobacterium vector. Why? For plant, vector is agrobacterium. For animal, vector is retrovirus. As simple as that. So using agrobacterium vector, these nematode specific gene are introduced in the host plant. These gene are introduced in the host plant. Introduction of DNA was such that DNA will make both sense and antisense RNA in the host. Correct? Because both the RNA are complementary to each other, it will form double-stranded RNA. And because there is double-stranded RNA, so RNA interference. RNA interference means harmful mRNA will be silenced. Harmful RNA of nematode will be silenced, right? Harmful mRNA of nematode will be silenced. What is the benefit? Now, parasite cannot survive in the transgenic host. Why? Because it will interfere nematode RNA. So in this case, plant is not affected by the parasite nematode incognition, as you can see. Double-stranded RNA trigger protection from the nematode infestation. First is the root typical control plant. B is the transgenic plant 5 days after the infection, but protected from the novel mechanism. In second case, there is a proper protection. In second case, there is no infection. Correct? It is 254. 254. I started at 2, right? 2 or 2:15?
So 2:15, 20.
Yes, sir.
2. So in next 35 minutes, I will finish this full chapter maximum. Okay. Come to the. We know RNA interference is important if we want to protect the plant from nematode using double-stranded RNA interference. Correct. Next, biotechnology application in the medicine. Recombinant DNA technology have lot of benefit because we can produce mass level medicine which are more safer and more effective. More safer and more effective. Further, recombination therapeutic are not inducing unnecessary unwanted immune response. Biotechnology product are not having the unnecessary response because normally when we take from the non-human anything and try to introduce in human being, then of course there will be immunological response. Correct? If we add pig pancreas in human body, then human body will not receive this. But here, no issue. Currently, we have how many medicine recombinant therapeutic? 30 in all over world. In India, 12. All over world, 30. India, 12. Right? Which is marketed. 30 all over world approved. 30 recombinant therapeutic approved for human use. Approved for the human use 30. And how many we are using? I think this is NEET 2025 question. Insulin. When we manage diabetes, it is possible only by taking insulin. Correct? But we'll realize कि when we isolate, like when we take insulin orally, there is a problem. What problem? Insulin is ultimately protein. When we take in the form of tablet, our body enzyme will destroy the protein, and this insulin will not be beneficial. Correct? So now we can make human insulin using bacteria because bacteria can divide fast and bacteria can produce lot of protein. Correct? The whole process will become easy. Why? Because large quantity of insulin bacteria can make. Correct? But can we give orally? Answer is no. Okay. Why? Because ultimately insulin is a protein. Digestive enzyme will break this. Correct? So insulin used for the diabetes, earlier we used to extract the pancreas of the slaughter pig or the cattle. Okay. But the problem is when we obtain insulin from the animal source, it causes some kind of allergy or some kind of reaction. Okay. When we study insulin, we get to know there are two main chain A and chain B in insulin, mature insulin, and both are attached by disulfide bond. Okay. Example, as you can see, one disulfide bond, two disulfide bond, and third disulfide bond present on the A chain. B chain is longer, A chain is shorter. So A and B, mature insulin. But in immature insulin or proinsulin, you have C peptide also. Correct? In mammals like human being, insulin is synthesized as prohormone, like proenzyme, inactive form. Correct? We need to process it before becoming fully mature and functional hormone. To make it functional and mature, we have to process it. Correct? Here, we'll have extra steps, and this extra step is C peptide. Extra step is C peptide. C peptide is not present in the mature one. Why? Only pro-hormone, pro-insulin have C-peptide. When you make it mature, active, you remove this part. Correct? The main challenge of producing insulin using recombinant technology was to getting insulin assembled in the mature. You can get chain A, you can get chain B, but how to assemble? So 100% it will act. In 1983, Eli Lilly company of the America prepared two DNA sequence, and these two DNA sequences were exactly same as chain A and chain B, correct? And they introduced this chain A and chain B in the plasmid of E. coli bacteria. Okay, E. coli bacteria produce a lot of insulin chain A and chain B, correct? Next, both the chain were produced separately. Chain A in different bacteria, chain B in different bacteria. Then we
So
extracted
can you
those
say
chain.
about the C peptide?
Yes, and we combined them by creating disulfide bond to form human insulin. सी पेप्टाइड इमेच्योर इंसुलिन में तो होती है, लेकिन जो फंक्शनल इंसुलिन होता है उसमें से रिमूव हो जाती है. तो इंसुलिन तभी काम करेगा जब उसकी जरूरत होगी, अदरवाइज वो काम नहीं करेगा. एनी डाउट?
Yes, got it.
If a person have hereditary disease, we cannot treat such disease. But when we read about the gene therapy, in gene therapy we use method to correct the gene defect. But we can do this only when we diagnose the disease in the child or early. Correct? How? Because normal gene will be inserted in the person's cell, normal gene, or tissue will be treated by using normal gene. Correct? Correct. So we can add in the individual or in the embryo normal gene, so there will be normal function. Correct. First gene therapy was done in 1990, 4-year-old girl, and she had AD deficiency. This enzyme is very, very important for the immune system to function normally. Correct. If we have disease, because ADA there is a gene which is responsible for adenosine deaminase. This gene will be deleted. Okay, in ADA deficiency, this gene will be deleted, which is responsible for this enzyme. In some children, what will happen? We can cure this ADA deficiency by bone marrow transplantation. One option is this. One option is bone marrow transplantation. In other, we can treat by enzyme replacement therapy. So if question asks, what are the possible options to cure ADA deficiency? Bone marrow transplantation and enzyme replacement. Correct? What happen in case of enzyme replacement? A functional ADA will be given to the patient by injection. But problem with both the approach is that both are not completely curable. Example, this is one of the first step in the gene therapy. What we'll do? Lymphocyte will be taken from the blood of the patient, culture outside. Functional ADA's complementary DNA. Here we use retrovirus vector. Why? For human being, we use retrovirus. For plant, we use agrobacterium. So we'll make complementary DNA and introduce this lymphocyte. Correct? and we make thousands of copy of the complementary DNA and introduce into the patient. But the problem is, after few days, this cell will die, correct? And parent periodic supply of this cell is really important. Okay? In every few days, like every month, you have to do the same therapy, correct? But the best part is, if we can isolate gene from the marrow cell that produce ADA and introduce into the cell at early embryonic stage. If we do same thing in the early embryonic stage, it will be a permanent cure. But it's difficult to do. In early embryonic stage, it's difficult to identify whether children have this disease or not. Embryonic, remember embryo, not even child, embryo. Are you getting my point? Practically, it is possible. Theoretically, it is possible, but practically, it is difficult to identify disease in embryonic stage. Can you imagine embryo? Right. Next. We know effective treatment of the disease, early diagnosis, everything is important. According to the old method, conventional method of diagnosis, what are the conventional method of diagnosis? Serum analysis or urine analysis are the old method. Correct? In those cases, early detection is not possible. Early detection is not possible. Correct? So, three technology, recombinant DNA technology, PCR, and ELISA. Using these three, we can use early diagnosis. We can use early diagnosis. Correct? If we see any pathogen, normally we can identify the pathogen whenever there is a disease symptom. अगर मुझे आज टाइफाइड हुआ है, तो जब टाइफाइड के सिम्टम्स होंगे, only then I can see कि okay, I am facing issue. I have to approach doctor and I have to get diagnosis done. है ना? But if we can do it in the early stage, so I don't get proper symptom, because by the time symptoms come, it's already late and lot of pathogen is already present in human body. Okay, lot of pathogen are already present in the human body. Correct? But we can identify very low concentration of bacteria by amplifying using the PCR. So imagine PCR made our job very, very easy. That's why PCR is used in the early detection of HIV. It can detect cancer in the early stage, correct? And it is very important to identify many genetic disorders in early stage. But for this, we need to get regular checkup done, even when we are okay, when everything is perfect. Give me 1 minute, guys. Hello. Okay, come to the topic. Very popular, autoradiography. Yes, Pratigya.
हेलो
Hello.
सर, आपने मुझे रिमूव कर दिए।
हाँ,
सर मैं आपको बोली 2 मिनट में आती हूँ, नेटवर्क किससे थोड़ा बोली मैं आपको।
2 मिनट तो मैंने कॉल किया उसके बाद भी मैंने बोला कि कौन कौन है यहाँ पे, तो नहीं आए।
सर मैं आपको बोल के गई थी, आप
फिर 2 मिनट से ज्यादा हो गए थे ना, क्योंकि इतनी देर अगर होगा तो फिर कैसे काम
मैं
चलेगा?
बीच में वीडियो ऑन की थी,
नहीं फिर
अभी थोड़ा नेटवर्क इश्यू हो रहा था, मैं क्या करूँ?
फिर ये
नेटवर्क थोड़ी मेरे हाथ में है।
जैसे ही नेटवर्क इश्यू हो तुरंत टेक्स्ट कर दो कि सर ऐसे ऐसे मेरा नेटवर्क इश्यू है।
तब मैं जस्ट कनेक्ट करने ही जा रही थी, आपको रिमूव कर दिया।
कर दिया, ऐड कर दिया।
सर आप ऑफलाइन छोड़ो, ऑनलाइन में भी इतना मतलब सोचा नहीं था।
ऑनलाइन में तो कंट्रोल करना और भी इजी है। बात यही है कि जो सीरियस स्टूडेंट है ना वही पढ़े। अगर कोई वीडियो नहीं करना चाहता तो मत करो, चॉइस है। एटलीस्ट हम ये तो ना कि करना ही है, मत करो। हु एंटर्ड? सारा। He is very obedient about the camera, so no issue. So come to the point. This auto radiography is very, very, very important, very popular question. Okay. What happened in auto radiography? We use single stranded either DNA or RNA. In this case, single stranded word is really important. If we can use both DNA or RNA is important. We also use radioactive. Yes, Barinder.
हेलो सर, क्लासमेट से लो।
हाँ, लेकिन कैमरा ऑन करना है। मैंने पहले पूछा था कि हाँ,
अभी पॉसिबल नहीं है
क्या हो गया?
सर, वो ना फोन चार्जिंग पे है और मैं उसकी कैसे करती हूँ,
हाँ, तो
वो नहीं हो पाएगा।
टेक्स्ट कर दिया करो कि सर ये इमरजेंसी है, इतनी देर में मैं कर दूँ, कोई दिक्कत नहीं, मैं तो समझता हूँ। तो हाँ
मैं
भाई।
बोलने वाली थी आप मेरे को कि फिर
You don't check messages, sir. That's a thing.
नो नो, आई विल चेक मैसेजेस।
I sent
कर
a
दिया था
message
यार,
11
कर दिया।
o'clock. You didn't see.
हाँ। Anybody can have emergency, but at least tell in advance. Next.
the problem is sir, you don't check messages like Jordan said.
What I can do, I get so many messages, so I don't know what to do.
The archive the restaurant keepers and important. That's it. Easy. What I've done is the same thing.
I don't block people, I don't archive people. So in future, if I get any important message, at least I'll get to know. Because I don't check archive. I have put more than 50 chat in archive. And if there is any important thing, I don't get the notification and I feel bad.
I've put everyone on archive except for my parents and our class groups. That's it.
Okay. In archive, there are 142 chat. Okay. In one group, there are 869 messages, another group
Sir, it's
999.
archive or archive?
Yeah, that's the thing.
What's the
It's
pronunciation?
archived.
Yeah, I've put so many things. I have put NMC, CTY, ITY, ITY.
You still part of those groups, sir.
No, but there was chat and I put an archive when I left.
So I missed those NMC group chats, sir.
Okay. So move to the next part. Photoradiography is really important. Here we use probe. Okay, radioactive molecule probe. This probe will bind with the complementary DNA and it can be detected by autoradiography. Clone having mutated gene will not appear in the photographic film. That's why probe will not have complementary with the mutated gene. As simple as that. How many of you did not understand this autoradiography properly? Raise your hand. ये सर ये समझ में नहीं आया अच्छे
So
से।
can you do the
वरिंदर।
molecular diagnosis again?
हाँ। So quickly raise your hand. How many of you don't understand this? थोड़ा सा तो ऐड कर लेते हैं बेचारों। वरिंदर, सारा, बबली, चंदना, रेयान। ओके। कैसा लग रहा आदित्य?
सर मैं 2 मिनट के लिए बाहर गया था, आपने निकाल दिया मेरे को।
अरे बता दिया करो, कोई दिक्कत नहीं है। ये बस इसलिए है कि बस थोड़े से बच्चे सीरियस रहें, जो भी रहे, है ना, कि काम हो सकता है बट बता दो पहले। बस मैंने थोड़ी सी देर रखा, ज्यादा लॉस नहीं करवाया, ये रियलाइज करवाने के लिए कि वीडियो ऑन करना जरूरी है। तो ऑटो रेडियोग्राफी समझ लेते हैं। एग्जाम्पल, यू हैव दिस ऑटो रेडियोग्राफी। ओके, In autoradiography, let's assume there are total 4 DNA. Okay. And we use probe which is radioactive. Radioactive means color. Radioactive means colored. Now what will
Radioactive
happen?
means it can be detected under like X-ray. So
Yes, color. Radioactive means color in the simplest form. So what will happen for most of the normal DNA? We know they're complementary. Example, this is not normal, this is abnormal, and remaining three is normal. For the normal DNA, we know they're complementary. They will bind and show color like this. Okay. First three are complementary, so they will show color. But last one, we cannot tell or we cannot design probe of the mutated gene because we don't know. Correct? We don't know their mutated gene, what is their combination. So abnormal gene will not be radioactive in nature. So it means fourth gene is mutated, first three gene are normal. Understood? That is autoradiography. Purpose to identify mutated gene. Principle, we have complementary of normal gene but not mutated gene.
so
Understood?
can you explain it again? So the ones which don't have the probe on it is considered abnormal and radioactive.
Yes. No, it is not radioactive.
Not radioactive, it's just abnormal. Okay.
Yes.
It
And
is
for normal,
radioactive.
because our probe can bind to them, probe is radioactive, so the complementary DNA will be radioactive, right?
So now one question, sir. Let us
Yes.
say that in that certain region there are so many cells with there's so much genetic material, the same number of codes, and there are chances of like in that millions there must be thousands of different variation. How will we know if it is abnormal
So
by
we
the
can
number?
have, like, we can develop thousands of probe. Probe is not only one type.
Sir,
Depend upon the
no, you'll
combination.
develop so many probes, you put on so many cells, but then there should
Um,
be like a number of abnormal genetic material to classify it
so
as
we can
cancerous.
identify all abnormal genetic material by
So
using
even
this.
if one is abnormal, that means there's something wrong with it, or what, sir?
Yes. Yes. Even if one is abnormal, it means something is wrong. Then based on that, we'll see how many abnormal.
So
So what is the impact of that?
100% radioactivity should be there for it to be normal.
Yes.
Got it, sir.
Last. Everybody understood, guys? Last is ELISA test based on the antigen antibody interaction. Antigen antibody interaction, because whenever pathogen attack, our body will produce antibody against it. So either you identify the antigen or you identify the antibody. If any of the thing is present, it means there was some pathogen. Example, in corona time, we used ELISA. Even people are normal, but they had corona symptom before, like not even corona symptom, coronavirus in their body. What will happen? We could identify this by using the antibody antigen method, ELISA method. We used a lot in case of this, in case of corona. Transgenic animal. Animal whose DNA is manipulated is transgenic, correct? And animal that have extra gene is transgenic. We produce transgenic rat, we produce transgenic rabbit, we produce pig, sheep, cow, or fish, correct? But majority are mice. 95% are mice. Why? Small size, less lifespan, easy to produce, metabolic activity is high. There are many benefits. Okay. Why we use transgenic animal? To understand the normal physiology and development of human being. Transgenic animals are designed to study how genes are regulated, how they affect the normal function of the body. Correct? Example, if you study the complex factor that involve in the growth of like human insulin-like growth factor, if you introduce gene from the other species, then factor will be different. There will be some biological effect. We can understand. Example, there is one gene responsible for increase in the height. If you remove that gene, what will happen? height will reduce.
Growth stops or growth reduces?
Yeah, growth reduces, right? So you
So
will
how
get to
can
know.
growth reduce, sir?
Example, you have total 30,000 gene in one DNA, like one cell DNA. If you remove one gene, and after removing this gene, height is reduced, it means this removed gene was responsible for the height.
Sir,
Make
but
sense?
growth is irreversible, है ना?
And in case of mice, we can study it fast now because they have very short lifespan. In the new
So, but
mice,
then I was just confused because like if height increases, there's no way to reverse
in
the.
new organism we can study. Example, I'll give you another example. Uh, there was protease enzyme produced by the one particular gene
Okay,
and
sir.
responsible for the protein breakdown. If you remove this gene, then protein breakdown will not happen. If protein breakdown do not happen, it means gene was responsible for protein breakdown.
Got
I
it.
gave this example to
Got
give
it.
idea. Second is the study of the disease. Animal are designed in such a way that we get idea how genes are responsible for the disease. Correct? So based on this, we specially design some model for the human disease because we want to study the human disease and we want to treat them. Correct? Today we have model for the cancer. We have model for the cystic fibrosis, for rheumatoid arthritis, for Alzheimer's disease. Okay, these are the transgenic model today we have for human being. Okay, so we can study the gene how they are responsible for the disease, biological product. Medicine, when we use, sometimes there are biological products, but they are very expensive to make. Using the transgenic animal, we can produce biological product at the larger scale. Correct? Example, human protein, alpha 1 antitrypsin. This product is used to treat emphysema. Using the transgenic animal, we can produce at the larger scale. Alpha 1 antitrypsin for emphysema treatment. Same way we used phenylketonuria treatment and cystic fibrosis treatment using the biological product. In 1997, we used transgenic cow Roji. This transgenic cow Roji have high protein, correct? This much gram of protein. And her milk contain alpha-lactalbumin and her milk was more nutritionally balanced compared to the normal milk than natural cow milk. Not we are not comparing with the mother. Of course, mother milk is best for the baby. We are comparing normal cow milk and transgenic cow rosy milk. Rosy milk is nutritionally more balanced. The rosy milk have more protein. Rosy milk contain uh, more alpha lactalbumin. Albumin means what protein? Right. So from transgenic animal, we can get product alpha 1 antitrypsin emphysema, and from rosy transgenic cow 1997 alpha lactalbumin and more nutrition. We use transgenic mice for vaccine safety. Before we use vaccine on the human being, we first use them on the animal. Correct. Same way, we use transgenic mice for the safety of the polio vaccine. When we feel ki it is successful on the mice, then we use on monkey to test the safety batch and then it will come to the human being. Correct. Then chemical safety testing or toxicity testing, safe testing. Is chemical safe for us or not? Okay, is chemical safe? Example, we are using one food. Is this food good for us or can it have harmful effect on our body? So we use same thing. The procedure is same used for the drug toxicity because we want to understand is drug really harmful for us or beneficial. Correct? So transgenic animal are designed in such a way that they are more sensitive to the toxic compared to the non-transgenic animal. Okay, so we can study that toxic substance easily. Correct? So we can get the better result in the less time. Let me tell you what. Normal mice or normal human can consume some toxin, still they are okay. They can absorb it, right? But when we make transgenic mice, they are oversensitive. Even a small amount of toxin when present in the food, they will show result. They will either die or they, uh, you can say, they will show some result, some disease condition. So you can study, ki, okay, this chemical is harmful, so we should not use this. You know, the real soldier in the world are actually mice because they are losing their life to um, get the better quality product for the human being. Still, we don't consider them real fighters. Ethical issue. Right? When we manipulate the living organism, it should be regulated. Otherwise, sometime, example, zombie movie you have seen, it can sometime lead to the unpredictable result that we can't even imagine. Correct? That's why morality is also important because when we are modifying the organism, many organisms are killed. We should not do this. That's why Indian government also set up a organization known as GIAC, Genetic Engineering Approval Committee GIAC. This committee make decision regarding whether we should do genetic modified research on particular organism or is genetically modified organism is safe for the public service or not. Example, you use a genetically modified dog and you feel that these dog can train easily and they are good for the human health. But later on, dog become aggressive and start killing the owner or people. Then automatically it is dangerous. So GIAC take decision that is it safe to introduce GM organism in the public service. Correct. When we modify the living organism for the public services, it can also create problem with the patent. You, example, somebody else take patent in the wrong way. Correct. So, example, when we take about the food or medicine source, so people can take patent, and this is the public anger. Many company um, take the patent, which is actually is not their actual research. Correct. Example. So, rice is the important food grain and we have long history. Asia have long history of the rice. Correct. Like we have 2 lakh variety of rice in India alone. Correct. India is one of the richest rice diversity country. Basmati rice is famous for the unique aroma and flavor. Unique aroma and flavor. And we have 27 documented variety of the basmati grown in India. Correct. And we have this basmati rice in the old text of folklore or poetry from the century. But in 1997, American company got patent right on the basmati rice through US Patent and Trademark Office. It means now only this company can sell new variety of basmati in US or abroad. Correct? So new variety of basmati actually is derived from the Indian farmer variety. So Indian basmati was crossed with the semi-dwarf variety and claimed that they invented the new plant. Correct? They invented the new basmati. So patent can be extended to the equivalent ki no other people can sell basmati rice without the permission of this company. Correct? Same attempt were made to use multiple products like traditional Indian herbal medicine like turmeric and neem. They tried to take patent for the turmeric and neem. So if we are not aware and we don't take immediate action for the patent, then many other country and people may can enrich our legacy and we can't do anything because they took patent. And in this case, we have one word biopiracy. When company use the resources of a country without proper authorization of the government. Okay, without any proper payment. Example, company is using the specific plant of India selling throughout the world, but they are not giving any money to the farmer or government. It means this is wrong. Correct. You will see most of the industrialized nation are rich financially, but poor in the biodiversity and traditional knowledge. Which country should I keep? Like Germany. Germany do not have lot of diversity. Most of the country UAE do not have lot of diversity, but they are rich. Correct. They have very less diversity. They have very less traditional knowledge. And opposite to this, most of the developing country and underdeveloped country are very rich in the biodiversity and traditional knowledge of the bioresources. Correct. So when we say the traditional knowledge of the thing bioresources, many advanced country can exploit by using the modern application, and people don't know how to save them. Right. That's why many countries are taking action to prevent the exploitation of this. Correct. Same way, Indian Parliament recently cleared Second Amendment. Second Amendment of the Indian Patent Bill. This second word is very very important. And in this Second Amendment of Indian Patent Bill, we consider the patent term of emergency provision, research, and development initiative. Understood? Chapter is done.
So in NCERT, we don't have about the rice genome project and human genome project.
Come again, Ryan.
Sir, I'll be back in 2 minutes.
Okay, Anvita. So we finished this chapter. Any of you can ask any question. Full chapter is done. So take just 5 minute gap because it's been long class. But and in the same rapid fire round, we will solve 30, 40, or 50 questions of this chapter.
Sir, I
Okay.
texted the same.
Yeah. Let me screen share of recording stop. Be here only. You can just array. First question. Aditya.
Sir, bacterium.
Eli Lilly company used bacteria. Jordan.
Yes, sir.
Following steps are taken. Involve the BT toxin.
1 minute, sir. Bachelor's degree. B is first, produces crystal.
If you take B first, Bacillus thuringiensis produce crystal that is insecticidal protein. Okay, after that.
Yes, protein, correct. Then comes the toxins exist as in inactivate. B E. Then C is alkaline pH. B E C A D. Option D.
Correct. Imagine this question is not just question. This is the full summary of Bt toxin. See what happened in the sequence wise. First, Bt have this insecticidal protein. Second, these insecticidal protein are present in inactive form. Third, there is alkaline pH. Fourth, due to alkaline pH, there will be conversion, and fifth, bacteria will die. Next, Manan. Fate of
Yes,
the DNA carrying the gene of interest transferred to the alien organism.
एलियन डीएनए का फेट क्या है? एक तो वो
Hmm.
इंटीग्रेट होगा सबसे पहले, तो फिर वो मल्टीप्लाई करेगा।
Hmm.
सर ई भी तो होगा, रेप्लिकेट करने की भी तो पावर होती है फिर उसमें।
Hmm.
रेप्लिकेट भी तो होगा, अगर जैसे हमने डीएनए डाला, एलियन डीएनए डाला हमने किसी
Hmm.
भी उसके अंदर,
Hmm.
सेल के अंदर, तो अगर वो सेल
Hmm.
उसके बाद रिप्रोड्यूस मतलब माइटोसिस या मियोसिस के स्टेज में जाएगा, तो वो डीएनए भी तो रेप्लिकेट होगा साथ में।
हाँ, पता
होगा
नहीं
कि नहीं, मैं
जरूरी
ये
भी
पूछ
होगा
रहा
या
था।
नहीं। जरूरी नहीं है होगा कि नहीं, लेकिन इंटीग्रेट हो भी सकता है, नहीं भी हो सकता है, मल्टीप्लाई हो भी सकता है, नहीं भी हो सकता है। हमने तो पॉसिबिलिटी देखनी है ना, मे और मे नॉट। ऑप्शन के हिसाब से ऐसे ही बन रहा है, क्योंकि दो ही ऑप्शन करेक्ट है, तो बी और सी तो पक्के करेक्ट है। वाई बी एंड सी करेक्ट? बिकॉज इट मे और मे नॉट हैपन, इट मे गेट इंटीग्रेटेड विद द जीनोम, इट मे नॉट, इट may multiply or inherited with the host DNA or may not. That's simple. Next, Anvita.
Sir, C.
Protoplast will fuse. Next, Chandana. Alpha 1 antitrypsin,
3, sir.
Cry 1AB,
Corn borer.
1AC,
C1, D2.
ballworm, and enzyme replacement therapy 2. Pratigya, incorrect. Any doubt? Pratigya.
सर आपने ये प्रिंसिपल्स एंड प्रोसेसेस का निकाला है क्या?
ये दोनों मिक्स हो गए तो कोई बात नहीं, वो भी तो रिवाइज हो जाएगा।
ओके सर।
नेक्स्ट, Rayan. What is X and Y?
So 1 minute. Option
X,
B. Okay.
X is origin of replication and Y is row. Correct. X is controlling copy number in DNA. Y is protein involved for the replication of plasmid because it is raw, and this is ORI site. Correct. Next, Barinder.
Yes. Sir A.
This all other are correct. Here it is used to deliver the gene of interest in plant, host cell, plant. Next, how many are there? Nirdila,
Yes, sir.
molecular size.
Sir, A है A.
Very good. Next, Aditya, Jordan, gene therapy.
Gene therapy is allow correct for defect gene. RNA interference is for cellular defense. ELISA is for DNA, sorry, diagnostic test for AIDS, and gelatin for suppression DNA.
Yes, Aditya.
A part सर A.
ये अर्ली कर सकते हैं। नेक्स्ट, अन्विता.
C.
Yes. Next, Pratigya. Gene silencing, RNA
Gene
interference.
silence, yeah.
RNA interference. Next, Sarah. Because lymphocytes are not immortal, right? So we need to do it again and again. Next, choose the most appropriate answer. Anvita.
So 1 second. Take me to the correct, no?
Hmm.
Then insulin used for treatment. Correct? Pro
सर सी
correct, sir.
थोड़ी होगा
Hmm?
सर सी तो गलत होगा
Sir,
हाँ,
E सही है, हाँ. E सही है.
So C is
किसने क्या
correct
पूछा?
now, sir. It was used for.
Yeah. All of them are correct, no, sir?
Oh, all are correct.
We don't have all of them correct option. तो C नहीं है. Some patient develop allergic reaction to the foreign insulin. Not
Foreign,
to
not insulin, like foreign cells.
yes.
Yeah, okay.
So based on the option, you can easily eliminate.
Yeah.
So, but then
Insulin
the
used to
pink thing,
treat
can you explain
diabetes.
again?
Insulin pancreas used to transfer, not the insulin.
So, but they
Pancreas.
still extracted. Okay, okay, got it.
Yes. We got from the cattle and pig, but pancreas.
Basically, it's not wrong. It's just, it's not that.
It's not wrong. It's like you have to identify which
Thick
is more relevant.
statement.
Yeah.
Yes, more relevant. Then, Barinder, when gene targeting involves gene amplification is attempted in the individual to treat the disease. It is
Sir, B, gene therapy.
gene therapy. Next, Ryan.
Yes. First one correct, second one correct, and third correct, fourth correct.
ACD.
ACD. Yes, sir.
Next, Barinder. Correct.
Yes, sir.
Nirjala.
Yes, sir. Not correct. The function it is linked to whether she correct the genetic engine. D option. D.
Yes. Functional insulin A and B chain. Yeah.
Sir,
What?
B होगा.
Hydro
So,
Not
disulfide bond.
Shoot.
Disulfide bond.
Mhm.
Disulfide bond. Next,
सर डी तो फिर
Manan.
सही था जो ऊपर वाला
हाँ, D
क्वेश्चन
सही
था.
था. Pro in man insulin synthesizes pro insulin. No, pro hormone, pro insulin. Next, Manan, RNA interference gene is silenced using double strand RNA. Then Sarah,
Sir, option A.
option A, vitamin A enriched. Next, Jordan,
activation triggers. It's alkaline pH of gut.
yes.
Sir,
Next,
something was about midgut, right? What was that?
midgut epithelial cell of alkaline pH of like these cell will be affected, will be destroyed. Next, Aditya, feature of genetic code allow bacteria to produce human insulin by this.
C.
Genetic code are nearly universal. अन्विता, देखते ही पता चल जाता है इसको कि जीन थेरेपी
हाँ
एडीए डेफिशिएंसी. नेक्स्ट प्रतिज्ञा, देखते ही पता चल जाता है डाइहाइड्रोजन डाइसल्फाइड बॉन्ड. नेक्स्ट
So give us some time to answer also, sir.
अरे make it fast. That's
No,
what
but
you need to
within
do.
1 second, how can we answer?
So you only are marking and answering, sir.
Yeah,
Nothing
that's what.
else.
Yeah, but you have to make it fast. That's
So
what I am
before we
teaching
Sir,
you.
read
but
the question,
We
only
need to
move
read
off.
the
yeah,
question, right?
सर
Yes,
नीट में भी 30 सेकेंड का मार्जिन रखते हैं, सर आप 1 सेकेंड भी नहीं लगाने दे.
but
Sir, at least give one or
Sir,
two
see, I'll tell you
sentences.
what, sir, you've done this PDF 1,000 times. That's why you know what questions coming up
no.
next. You'll mark off and you'll
No,
go.
Even I make mistake. But I am
Sir, it's
telling
okay
you.
if you make
Example,
mistake, but you know the
listen
questions.
carefully. Wait.
But at least we need time to read the question, sir.
Yes, I understand. I do agree with your statement. But the point what I am trying to teach you, even in NEET, out of 90 questions, 30-40 question will be very lengthy. That might take 1 to 2 minutes. There will be some question in which you are supposed to do in even 5 seconds. Because
We're not
example, I
giving
have
5 seconds also, no?
Slow and steady wins the race. Slow and steady wins the race. Please don't rush too much also.
no. So what happened in this case? When you saw ADA deficiency, you know gene therapy only.
हाँ तो हम सर हमें बोलने दो ना वो जीन थेरेपी.
Let us say you will not let the other person answer, sir. You're just like, हाँ, ये तो answer है, ये तो answer है. And then you keep scrolling.
Correct. And the reason because I want you to teach ki how to make it fast, but what you guys said makes sense ki at least some time to read the question. So I'll give time to read the question. Okay. Next. Tobacco plant is infected by Melodygyne incognitia. Which part? Now correct? Yes. This is enough time?
Yes, sir, more than enough.
Yes. Golden rice, genetically modified in crop biosynthesis of what? Vitamin A. Next question. Anybody can answer now because we have to make it fast. tDNA into plant involve infection of plant by the Enterobacterium infestation. Next. Active
Catch off
because of
guard.
the
And see,
थी.
government of India
But
have one GEAC,
option A.
GEAC. This time is good enough?
Yes.
Yes, sir.
Yes, sir.
Fine, sir.
Yes. Crop engineer for the glyphosate resistant to this
He said,
tolerant to the what is this glyphosate? Even
So will it
they
be
must be
insects?
tolerant to the resistant to the insect. Only this look is herbicide.
besides I told.
Outer cellular.
Glyphosate is a broad spectrum herbicide. Okay, I don't know this. Glyphosate is the broad spectrum herbicide. Okay, which kill broad spectrum leaves herbs. Okay, crop plant may be affected by the herbicide. And now new crop are engineered for the glyphosate resistance. So phosphate herbicide is applied.
Old NCERT.
Yeah, this is from the old NCERT. Only weed and no other crop plant get harmed. Okay. I thought ki it is some kind of chemical present in insect, but glyphosate is the herbicide. It's like even after a long time, I'm reading in the same way. So
Option
I am
A.
almost equal
Option
to
A.
you.
Option A.
A.
Yes.
Insulin.
Next,
Cotton, sir, cotton.
Cotton.
BT cotton. RNA interference involve
Interference of
B.
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 complementary RNA.
Yes, sir.
Replace the defective gene in the therapy vector in the gene therapy is
G R I
D.
AI plus.
Cosmet.
A, sir, A.
Hey, adenovirus, we learned this yesterday.
because adenovirus infect human being. So
Yes,
we
sir.
can use as vector. Whatever affects
तो
human
भी
being,
क्वेश्चन था ना सर जिसका आंसर एडिनोवायरस था
It's
वो
rapid
रेस्पिरेटरी
fire.
इन्फेक्शन, रेस्पिरेटरी इन्फेक्शन के लिए बोल, वो क्वेश्चन पूछा था ना किसी से,
हाँ
साहिल शर्मा से.
फिर साहिल
Next, consumption of which of the following food will can prevent blindness associated with vitamin
So,
B deficiency?
golden rice.
Golden rice.
right.
Tobacco plant resistant to the nematode have developed by the introduction of DNA that
both
produce both.
sense and
Both sense and antisense.
Yes. Then it is a form of the DNA fingerprinting. Satellite
Option D. Satellite DNA
option
cause highly.
satellite DNA occur at highly repeated short DNA segment. कि satellite DNA repeat multiple times. I will explain this topic. Don't worry. First clinical gene therapy was done.
For ADA.
ADA.
D
Maximum number of existing transgenic animal are
Nice.
mice.
Process of RNA interference used in the development of plant resistance is
nematodes.
Nematode. Bacillus thuringiensis form the protein crystal which contain insecticidal protein. This protein
A.
bind to
A. It binds and causes lysis.
yes. Silencing of mRNA using the transgenic animal resistant to
Nematodes.
nematodes. First transgenic buffalo roji.
So no, cow is
वो
wrong.
Restriction enzyme are used in isolation of DNA from the other macromolecule.
Sir, A or D.
They're asking what is false. A and
D.
ADD. Disarm pathogen.
A, B is wrong. Downstream process correct. Disarm pathogen are used in the. So I think it should be A, B.
A and D.
A and B.
A,
A and
B.
B. Why? Because restriction enzyme to cut DNA, not to isolate from other
Oh,
RNA.
yeah.
or protein.
Isolation means everything else except for DNA. Yes.
GM brinjal in India developed for insect resistance, like same BT cotton.
BT cotton, the same gene there.
Same way. Everything same. Genetic engineering successfully used for producing
A part.
transgenic mice for testing polio vaccine before use in the human being.
So why not B, sir? Could you just show option B once?
Transgenic model for studying new treatment for certain cardiac disease, not certain cardiac disease.
It's okay,
Yeah. Certain disease is correct, but not cardiac disease.
Cardiac, okay, sir.
Some of the characteristic of the BT cotton are
Sir, option
C.
C.
correct.
No, wait.
Bollworm, na? Not diphtheria pest. Bollworm.
Oh, yeah. Diphtheria pest is for a cry.
Okay.
Bollworm, not for the other.
Yes.
Improved variety of transgenic basmati rice.
B.
The B.
Yes.
B, sir.
Commercial produced biotechnology procedure.
Option D.
Insulin.
Insulin. True about BT toxin.
Option D.
D. D.
D.
Yes, D.
Code दो sir अब.
Transgenic plant are the one.
So A.
A part.
A
A.
option.
BT is widely used in contemporary biology as
A.
A.
Yes, sir.
Yes, sir. Yes, sir.
antisense technology is used in
So what is antisense
Sir,
technology?
D. नहीं हाँ D no.
Option A.
A.
Option A. A.
Option A, RNA interference.
RNA interference, antisense and
Sense
nonsense
antisense.
codons basically are used here so that the genes
Yes.
stop sensing.
Yes, RNA interference, sense antisense. Cry1 endotoxin obtained from bacillus is effective against
Ball.
Bollworms.
Onboard.
Bollworms. Bollworms.
So
bollworm.
cry one, but B, right? Cry one B is for.
They didn't mention one AB or one AC. One thing.
Oh,
Second, they didn't mention corn borer option.
Sir, if it had option of convert, then
वो ऐसे नहीं देंगे.
okay.
उन्होंने इसी वे में डिजाइन किया है कि कंफ्यूजन हो, लेकिन फिर कंफ्यूजन ऐसा ना हो कि
Option
उनको
B.
क्वेश्चन
Option
याद
B. Golden rice. Golden rice. Vitamin A.
है. नाइट ब्लाइंडनेस, गोल्डन राइस, विटामिन ए. मेन ऑब्जेक्टिव ऑफ हर्बिसाइड रेजिस्टेंस GM crop is
Reduce
एक
herbicide
होता है
accumulation in food for health and safety.
C.
Hey, how, sir?
main objective of herbicide resistant crop is herbicide resistant is because we want to eliminate the weed from the field without using any manual labor. Because if you use herbicide resistant GM crop, you can kill weed directly. You don't need you to remove the weed.
So, but
Correct.
can you explain why B is in? I'm confused now. Little bit.
Logically, B statement is correct, but not with respect to the question.
so the
Main
main
objective,
objective is C, no?
main objective is to eliminate weed without manual labor, because now we don't need labor to remove all the weed. You just spray one this herbicide resistance GM crop. You don't need to use herbicide, and no weed will be there.
Got it, sir.
Actually, in this is 2025 NEET question also. They just wrote bacteria.
It's E. coli.
I think this answer is Pseudomonas putida.
Pseudomonas. I remember doing this, sir.
But this is not given in NCERT.
No, it's
Pseudomonas
not.
putida is used in the bioremediation process where we clean the contaminated site
So
using micro.
Xanthomonas is some colored wala, no, sir.
That is xanthophyll.
Sir.
Xanthomonas is the bacteria. Golden rice.
Elevation for vitamin A deficiency. So option B.
BT strain have been used in novel.
Bioinsecticidal plants.
More than enough, guys. Let's do last question.
D.
Option B, sir. Mechanism, gene
Option
regulation.
B.
Yes.
Identical. No.
Yes.
Oh, yeah, sir. Because
Because
improvement
genetic code is universal. If you have
universal in D. Yes, sir. I said
if
D.
you have GAG, whether it is bacteria or human being, GAG code for the same thing. AUG code for methionine always. Doesn't matter which organism. Right? So Jordan, was it really fast? Like what is required?
Very happy.
So that is the benefit of reading chapter. Now, if you get question from insulin or like RNA interference or BT cotton, you can make it fast. Golden rice A,
Yes,
right? Vitamin A. So it will be easy now because we solved 60 question in just I think 10-15 minutes. Same thing happens in NEET exam. Only problem is you cannot scroll. So you need break.
सर लंबा ब्रेक दे दो सर थोड़ा.
लंबा ब्रेक तो फिर इवनिंग में देंगे ना. ये अभी
इवनिंग हो जाएगी सर 4 के बाद.
सर
इवनिंग
मुझे 6 बजे चाहिए तो मैं भी पढ़ लूंगी.
नेक्स्ट चैप्टर how many chapters are left from the 12th class?
11th we haven't done much, so we need to like start, I guess.
सर 11th में reproduction unit बचा है ना
11th में reproduction
11th
unit?
sorry
reproduction.
12th my bad
Reproduction unit we need to do. So
Sexual reproduction we have done now for both plants as well as humans. Reproductive health,
we
I think we
covered
need to
many
do.
topics, not fully.
Not fully, sir. And
Yes.
reproductive health we need to do.
Yes, reproductive health we need to do.
सर ecology आपने करा पूरा नहीं ना but I feel like ecology से पहले हमें 12th के human physiology वाले chapter करने चाहिए
We'll do next reproductive health, small chapter, we can make it fast.
yes sir
Correct. So
सर अगली क्लास कब है?
7:30 रखें सीधा.
हाँ सर
So till then, if you feel any of your chapter is weak, you need to revise again what we did. Do that properly. Okay. See you then at 7:30. Which chapter should we do? We will do 2 chapters. Can we do one reproductive health and one organism and population?
okay sir
यस सर.
2 small
Yes,
chapters.
sir.
We can make it fast. Correct. So by that we'll be done with many of the things. Biotechnology unit done. This human health and microbes in human welfare done. Evolution done. Genetic many part is done. So we'll see you at 7:30. Bye guys.
Bye.
बाय सर.
Okay.
thank you thank you sir
Okay. Okay.